Thank you all for coming. As many of you know, recombination rate tends to vary across the genomes of most species studied. These pictures here show human chromosome 1 and Arabidopsis chromosome 1 recombination rate on the y-axis. Recombination rate is often low near the centromere, higher outside, and sometimes very high near the telomeres. For over 20 years, we've also known that recombination rate tends to correlate with the abundance of nucleotide variation, often as measured using pi. This is some data we have from our lab from Drosophila pseudoobscura, where the x-axis is recombination rate, the y-axis is variability. You see this positive association. Specifically, low recombination rate seems to be associated with reduced diversity. It's long been suggested that natural selection is involved in this, but the type of selection has been uncertain. Two prevailing hypotheses are selective sweeps and associated hitchhiking, hence the thumbs up, or background selection. So let me go over each of those in turn. Imagine each of these lines indicate a haplotype across a chromosome, with the little tick marks on the line indicate allelic differences relative to a reference. So you see the same variation here in a region with low recombination, a region with high recombination. If we add an advantageous mutation, it will start to spread, and as it spreads, it drags with it variation at nearby nucleotide sites. However, in regions of high recombination, the adaptive mutation will dissociate from the original haplotype, and you'll see the spread of multiple haplotypes. Whereas in low recombination, you have a single haplotype spread, and all variation is eliminated. In contrast, background selection, rather than using advantageous mutations, is using a range of deleterious mutations. What this does is it dooms that haplotype to eventual loss. But again, as with selective sweeps and hitchhiking, you can dissociate the haplotype from this detrimental allele. So for example, focus on this one over here, where some haplotypes will actually be able to persist in regions of high recombination that weren't able to do so in low recombination. So again, you have this overall positive association. It's hard to distinguish the effects of sweeps from background selection. Repeating background selection is BGS. Now sweeps have been observed, often because they have dramatic effects. With a classic hard sweep, you're introducing a new allele, and it spreads. And after it spreads, you're creating a fixed difference, either relative to other species or possibly relative to other populations within species. With background selection, it's a lot more subtle. Right? You have these new variants introduced, and then they're eliminated, and it doesn't really create anything. So how do you test it? Now, a lot of studies have seen patterns consistent with background selection, but they're often conflated with the same predictions you'd see from sweeps. So what my student Catherine did is she focused on a subset of genes for which there were no opportunity for recent classic hard sweeps. So these are genes for which there's no fixed differences between two closely related species, Drosophila pseudoobscura and Presention. So our prediction is that high conservation, so basically high similarity across a long time, should indicate there's been more negative selection. As more alleles are introduced, they're immediately eliminated or eliminated fairly soon, and therefore more background selection. So you should see low pi in genes that have low KKS, or low pi in just genes that have just overall low divergence. And similarly, there's the classic prediction that background selection should be more effective in regions of low recombination, so low recombination rates are associated with low pi. So she tested this in Drosophila pseudoobscura and Drosophila miranda, and the results are pretty clear-cut. Here's the relationship of KKS to pi. The blue line indicates Drosophila pseudoobscura, KKS is on the x-axis. There's absolutely no association. P is 0.93. For Miranda, also non-significant. When you look at divergence, there's a slight trend, but nowhere near statistical significance. And again, you do not see this reduction in pi associated with reductions in divergence. With recombination rate, maybe a hint of something in Drosophila pseudoobscura. You do see some reduction of variation in regions of low recombination, but it's barely significant, and nothing in Drosophila miranda. So again, these are very conservative tests, and they're only looking at eliminating the effects of classic hard sweeps. It's still very much in progress, but generally speaking, she does not see any evidence that high conservation is associated with more background selection and therefore a noticeable effect on nucleotide variation. And very weak evidence that variation is reduced in regions of low recombination when you exclude that subset of genes that may have had classic hard sweeps. So overall, tentative conclusion goes with the thumbs up rather than thumbs down. That patterns of nucleotide variation in these species seems to be predominantly affected by sweeps rather than background selection. So I should acknowledge that Catherine Cronus, who's depicted here doing her thumbs up, was the one who did all the analyses presented here. Thank you.